So right now, I'm very excited to introduce Fayal Abderemane-Ali, who is currently assistant professor at David Geffen Medical School at UCLA. Before that, he did his PhD at the University of Nantes at the lab of Gilles de Luzon. I'm not pronouncing this well, but as a part of his PhD, he spent a year as a Fulbright scholar in the lab of Daniel Miner at UCSF and went on to do a postdoc in Daniel's lab afterwards. And he also trained in cryo-EM at SLAC in Stanford. I will just mention two awards that Fayal got, which is Paul Cranfield Postdoctoral Award from SGP, so very fitting. And also he was awarded for Best Talk at GRC Ion Channel in 2018. So I'm very excited to hear this talk. His research focuses mainly on voltage-gated sodium channels, sort of two main aspects, how they form molecular complexes and how to target specific isoforms of these ion channels. Okay, so without further ado, over to Fayal.
All right, thank you, Jelena. Hello, everyone. So before I start, I would like to take a moment to thank the EpIC organizers for putting up together this great platform, as Jelena mentioned, that promotes senior postdocs and junior faculty. And I've attended many of the previous talks, and they were amazing. I really enjoyed them. And I also appreciate the invitation to be a part of this. As I started my own group recently at UCLA, it was last year. I don't have a complete story for my lab yet. So today I will be telling you 3 stories briefly about my postdoctoral work in the lab of Dan Minor at UCSF. And this, the first story will be about how to survive a toxic environment and what we've learned from poisonous birds and frogs. The second story will be a brief story about the discovery of a unique sodium channel modulator. And the last one will be about the first structural view of an ion channel biogenesis, and this will be mostly on calcium channels. And for the first story, I would like to start by showing you these very nice pictures of poisonous animals. So as you know, many animals carry toxins as a way to defend themselves against predators. And here I'm showing some nice example. On the left, we have the pufferfish or fugu. This fish carries tetrodotoxin or TTX. And this fish is actually very popular in Japan because it's considered as a food delicacy. And some people enjoy eating it with some of the toxin in there, just enough to cause some tingling and numbing in your mouth, but not too much to kill you. Here on the center, we also have this butter clam, which carries STX or saxitoxin. And this is a toxin that was actually very popular in the 60s because this guy here, Gary Power, was a CIA agent. He was a pilot. And when he was caught in the Soviet Union, he was carrying this coin. And inside this coin, there was this pin that contained STX. And he was actually supposed to use it to commit suicide when he gets caught so that he doesn't give out government secrets. Obviously, he was smart enough not to do that. And so when he came back, that's how the story came up. And on the right here, we have this beautiful species, the Hulipirihui from Papua New Guinea and the poison dart frog from Central and Southern America. And these two species carry BTX or batrachotoxin. And this is actually the toxin that's used for hunting. So Native Americans will make poison darts and touch the skin of this frog, and that's enough to make the frog, to make the dart poisonous, so they can use it for hunting. Now, all these toxins have something in common, is that they target a family of membrane protein called voltage-gated sodium channels, or NABs. And as you all know, these are very important ion channels, as they are responsible for generating and propagating action potential in numerous tissues, including the brain, the heart, and skeletal muscles. And these channels are also the targets of many other toxins, but also drugs that I will talk about a little bit later, local anesthetic, anticonvulsant, and antiarrhythmics. So now the question is, when I was doing my postdoc, I was interested in the questions on how these animals survive their own toxins, because when you think about it, all these animals carry these toxins, and these toxins target ion channels, sodium channels, and these animals also require these sodium channels to function properly. And why don't they kill themselves? And these are questions that many other groups and people have been wondering, asking about. And here I'm gonna tell you the strategies. There are three main strategies to survive a toxic environment. And I will discuss one of them in detail. So the first strategy is called enhanced detoxification or elimination. And this is basically what happens when we drink too much alcohol. Our liver breaks it down and our kidney get rid of it. But this is clearly not a strategy that these animals want to use, because the whole idea of having the toxin is to be able to keep it intact and use it to defend themselves against predators, or use it to catch prey in some cases. So the second strategy is called target mutation. And the idea here is that the host animals will mutate its gene encoding for sodium channels so that the channel does not bind or rebinds with a much lower affinity, the toxin, so that the host animal can be safe. And this is a strategy that has been shown to be very effective in many cases, including the case of TTX and STX resistance. And here I'm showing a very nice example for this myarenaria, which carry STX. And this myarenaria has this mutation. This is the selectivity filter, and all sodium channels have this glutamate here, and this myarenaria has this aspartate. And this mutation was enough to cause a 3 order of magnitude reduction of the affinity of the channel for both TTX and STX, which is quite amazing when you think about this massive 2,000 amino acid channel that only one single amino acid change will have such a big impact. And recent structural studies actually showed how this works. Basically, this glutamate interact with both TTX and STX, and by mutating it to aspartate, you shorten the side chain, and that's enough to lower the affinity for the channel for TTX and STX. Now, the third strategy I would like to talk about is this toxin sequestration. And here, the idea is that the host animal will develop means to sequester the toxin so that it doesn't reach the channel in the first place. And that's also a strategy that we think is happening in this bird and frogs. And I'm going to show you some data supporting that hypothesis. So I started working on this project with Jack Dimbaker, who is ornithologist in the California Academy of Sciences. And Jack is actually the person who discovered for the first time that birds can be poisonous, and that he discovered that this bird from Papua New Guinea, the pirihui bird, carries batrachotoxin. And there's actually a very interesting story on how he discovered this, because he was working on the field, he caught the bird, and the feather of the bird opened up a little bit his skin on the finger, and he was bleeding a little bit, and then he had this reflex of putting the finger in his mouth. And he started feeling some pain and numbing and all some weird stuff happening in the mouth. And that's how actually he found that there was something abnormal in that bird. And he, of course, he didn't know in the first place what was it, but then he took some sample and did more analysis and found out that this bird has batrachotoxin, the same toxin that's found in the poison dart frogs. And so I started working with Jack, and we were able to clone and determine this gene sequences for NaV1.4 and NaV1.5, so the cardiac and skeletal muscle isoform for this bird. And then we decided to go and characterize them, these channels, and see whether they are responsive to BTX or not. Before I show you the data on this from the channels in the birds, I want to show you what happens to the human sodium channels when BTX is around. And here is an example of a trace of where the channel can open and close or inactivate. And when BTX is around, the channel can close, but it can no longer inactivate, at least not completely. So this is the first signature of BTX sensitivity. And the second signature is when you look at this voltage dependence of activation, this activation curve. You can see that when BTX is around, there is this leftward shift of this activation curve, showing that BTX is promoting or facilitating channel opening. And this is the second signature of BTX sensitivity. Now, when I did this experiment with both the cardiac and the skeletal muscle sodium channel of the bird, here I'm showing the data for the NAV 1.4 from the bird, I was able to see that these channels are just as sensitive to BTX as the human channels. You can see the incomplete inactivation here and the leftward shift in the activation voltage dependence. And that was a big surprise because we were hoping that we will see resistance in this channel that could explain how this bird survive their own toxin. And so while we're working on this, other studies were interested in these questions. And there was this study that showed that there are multiple mutations in the poison dart frog genes that could explain resistance to their toxin, to BTX. And these are genes that were sequence that were determined from sample from the poison dart frogs, the Phyllobatis terribilis. And there were about 5 mutations that were suggested here to play an important role. And a year later, this study showed that among those 5, there's only 1 mutation, this N2T in the segment 6 of domain 4. This is in the pore region of the channel. This study showed that this is the only mutation that was able to provide resistance to BTX in a rat NAV 1.4. And but there were some things that were not clear in my mind when I was looking at the studies, in the sense that, so first of all, the bird nav channels are BTX sensitive. So that was something that I was trying to wrap my head around, because at the same time, one could think maybe the bird and the frog have different ways of dealing with the toxin. So that was not a big issue for me, but it was quite surprising to see these differences. But the point that was unclear to me was that this N2T mutation was shown that it occurs with a very low frequency among wild Phyllobatis terribilis. So if most of the terribilis don't have these N2T mutations, how do they survive their BTX? And the third point was that this N2T mutation was actually completely absent in another frog carrying BTX, which is Phyllobatis orotenia. And so from there, we're wondering whether there are some alternative strategies of autoresistance in both the bird and the frogs. So I decided to get these frogs into the lab and work with them. And this was one of the best part of my postdoc, because these frogs are actually very beautiful, and getting them into the lab was very exciting. Actually, everyone in the lab gets super excited when I receive them. So they all come take pictures, and then I have to stay there to tell them not to touch them. But it was very exciting to work with these tiny frogs. And so what I did was I harvested some samples, the brain, the heart, and skeletal muscle. Today, I'm just going to tell you about the data about the skeletal muscle, where I was able to clone the full-length sodium channels, the full-length gene encoding for the sodium channels of these frogs. And the first thing, which was not a surprise, was to see that the N2T mutations was not there. But again, this mutation occurs with a low frequency, so it's not a surprise that it was not there. But the other thing was that there was more than 90 amino acid differences between the gene encoding for this frog's sodium channel and other sodium channel genes. And so the idea, or the hope at least at that time, was that one of these 90 amino acid differences may provide some resistance to BTX. And so I had to go through like a lot of sequence redesign because these genes are pretty unstable, but ultimately was able to get the right sequence, gene sequence, nucleotide sequence that gives the same amino acid sequence that was able to get the full length sodium channel for this frog. And we had gene synthesis and did the experiment, the same experiment I showed you earlier. And here again, I could see that these channels from this frog are very much resistant to BTX, incomplete inactivation, and this leftward shift in the voltage dependence of activation. And this was a very big surprise as well, because now we have the bird that carries BTX and have sodium channel that is sensitive to the toxin. And again, and here we have the frog that has the toxin, a lot of toxin, and that has channel that are sensitive to the toxin. And just to put things in perspective here, so it's probably clear. This frog is about 1 inch, 2 inch long, and it carries enough BTX to kill 8 human adults, which is a lot of toxin when you think about it. And so how can this little frog has so much BTX and also have channels that are resistant, that are sensitive to the same toxin? Also, another experiment I did was to add the N2T mutation on that channel. And here again, that didn't change anything about sensitivity to the BTX. And so from here, we hypothesized that there must be some things that something else other than target mutation. But first, we want to make sure that the frogs I cloned the channel from were actually resistant to the toxin, because these frogs were raised in captivity. And something I forgot to mention, also very important, is that these frogs get their toxin from their diet, just like the bird. And so if they're in captivity and they're not eating food that has the toxin, maybe they don't need to develop any resistance to the toxin. But here, the experiment was to see whether these frogs I cloned the channel from were resistant or sensitive to the toxin. Here I'm showing the control experiment, where the idea was to anesthetize the frog. Here, this is the Xenopus laevis frog that doesn't have carry any toxin. So the gray area is the anesthesia, where the frog is asleep, and then I would inject either PBS as a control, that's the black trace, or BTX. PBS injection, actually, you can see that the frog can wake up after about 30 minutes, and then it's fine. But after BTX injection, the frog wakes up earlier, and then ultimately dies. And this is actually something that we later demonstrate why this is happening, because BTX is actually a sodium channel activator, and the anesthetic agent, which is strychnine, we're using the sodium channel blocker. So there is competition between the two, which explains why the frog wakes up earlier. But this is typical of BTX sensitivity on the frog. Now, when I did the same experiment, on the Phyllobates terribilis, which is the poison dart frog, I could see that here there is no change at all. Whether it's BTX or PBS, the behavior of the frog was the same. The frog was just fine and could wake up and live this life. And this suggests that these animals are very much resistant to BTX, yet they have sodium channels that are sensitive to that same toxin. And I also did the same experiment using the other toxins I mentioned earlier, the STX and TTX. And here again, the Xenopus laevis was not able to survive any of these toxins, whereas the Phyllobates terribilis was able to make it, and they will wake up and be okay later. And so from here, we hypothesized that these animals, the frogs and also the bird, use a toxin sequestration strategy, which basically means that they can sequester the toxin. And so even though they have sodium channels that are sensitive to the toxin, it doesn't matter because they have ways to stop the toxin from reaching the sodium channels. And to test this, we turn into, this is just as a proof of concept, we turn into a toxin binding protein called saxifilin. And saxifilin is actually has been characterized in the 90s as in the American bullfrog, and that was shown that it can bind STX. And the structural information between saxifilin and STX was later on determined in the minor lab, where this saxifilin can bind, STX can bind to this tiny pocket here of saxifilin. So the experiment I wanted to do was to see whether saxifilin was able to rescue sodium channels that are blocked by STX. And here's the data where the experiment consisted of expressing this Fayal stereobilus NaV1.4 channel. That's the black trace. You have the activity channel can open and inactivate. Now, when I apply 100 nanomolar STX, I could block 90% of the channel, which was expected. And now when I add saxifilin on top of that at a 1-to-1 molar ratio, I was able to see a 50% recovery of channel activity. And this recovery was actually 100% when the molar ratio was 2-to-1. And so what this means is that saxifilin can effectively rescue channels that are blocked by STX. And what was amazing also was that how fast this recovery was happening, because you can see that 100% of the channels were able to be recovered in less than a minute, which was quite impressive when you think about the very high affinity of the channel for this STX. And so back to our question, because now we have this proof of concept that this sequestration is a very efficient mechanism to protect channels from against toxin. I just showed you how saxifilin is a toxin sponge protein. And we think that in the case of the bird and the frogs, there is this sequestration strategy that's being used to protect the animals. And we hypothesize that there's a batracofilin, which would be another toxin binding protein to protect these animals against BTX. And it was actually very exciting to see the reach of my work as it got picked up not only by mainstream journals, but also popular magazine. It was even more exciting to see that these recent studies from Lauren O'Connell's group that show that an alkaloid, a plasma globulin, is able to bind and sequester a poison frog alkaloid. This was on another toxin or another frog, which is a toxin was called pimiliotoxin or PTX. But it was very exciting to see that this strategy holds, and this is something that's being used to protect many other species against other alkaloids. So that was quite cool to see. And my lab is also interested in contributing in these questions about autoresistance for many other toxins. There's over 800 alkaloid toxins out there, and I think it would be very cool to study them and understand how these animals survive these toxins. And this can also help to design new antidote or molecules to protect ourselves against these toxins. Now, I would like to switch to the second story and briefly talk about the discovery of a unique sodium channel modulator. And so this story actually started as a collaboration with the Justin Dubois lab at Stanford. And here I'm showing, again, the trace of the human sodium channel with activation and inactivation. This is a very, very simplified kinetic model where the channel can close, open, and undergo either fast inactivation or slow inactivation. And today we know a lot about closed, open, and fast inactivation, but we know very little about the conformational change and residues involved in slow inactivation. And the slow inactivation is actually very important because when it's defective, it's involved in many disease. And here we took advantage of BTX effect on inactivation to basically study how it works, but also to see whether we could find some nice derivatives with interesting biophysical properties. And so this work was initiated by Tim McKenzie, who is a very talented chemist back when he was a grad student in Justin's lab at Stanford. Tim was not only able to make BTX, but also make many derivatives of this BTX. So today I will show you the data only on 3 of these derivatives. So this is the parent compound, which has this dimethyl pyrrole here in the C20 position. And Tim was able to replace this pyrrole ring with the benzoate, and that's how he could get this BTXB derivative. And when this was replaced by a cyclohexane carboxylate, he was able to get this BTX-CHX, or completely remove the ring and put a heptanoate there. And so that's how he was able to get this BTX-INE. So all these compounds, both the BTX, BTXB, BTX-CHX, and BTX-INE, are able to cause this leftward shift in the activation voltage dependence, showing that they are acting just like BTX, they are promoting channel opening. What was interesting is looking at the effect of this compound on the channel inactivation. And how we assess inactivation is basically depolarize the membrane at different potential, and after each potential, we'll step again, depolarize at 0 millivolt for 100 milliseconds, and see how many channels are able to fire again. So we are looking at channel availability as a way to assess voltage dependence of inactivation. And we can actually either assess overall inactivation or fast inactivation or slow inactivation by changing the time of this pre-pulse depolarization. And here on overall inactivation, you can see that the control condition channel without any compound can undergo this voltage dependence inactivation. The more you depolarize, the more you inactivate the channel. And when you apply BTX, BTXB, or BTX-CHX, there was no inactivation at all. But when you look at the blue trace, BTX-INE, we could see that there is inactivation occurring in this condition. And that was very interesting to see, because this was the only compound that showed this behavior. Now, when we look at fast inactivation, by reducing this pre-pulse duration to 10 milliseconds, we could not see this inactivation occurring with BTX-INE anymore. However, when we have a longer pre-pulse, this time 6.5 seconds, we can see again that this BTX-INE is able to allow some inactivation to occur. What this suggests is that this BTX-INE is able to block fast inactivation, but not slow inactivation. And that was very cool to see. Now, the ultimate test was to see on the channels that does not have fast inactivation. So we turn into bacterial sodium channels. Why bacterial sodium channels is because, so on the eukaryotic sodium channels, we have the monomer of 4 different domains that are similar but not identical. And there is this IFM particle that's between domain 3 and domain 4 that's responsible for fast inactivation. In bacterial sodium channels, we have homotetramer. Therefore, we don't have these IFM particles. Therefore, these channels actually do not undergo fast inactivation. So only slow inactivation occurs on these bacterial sodium channels. So testing this compound on the bacterial sodium channels, we're able to see that they all cause a leftward shift in the activation voltage dependence. So there was no surprise. And here we can see that when we look at inactivation, BTX, BTXB, and BTX-CHX block most of the inactivation, more than 60% of inactivation in these channels. However, BTX-ion has only a very little effect. So basically, inactivation can occur almost as much as in the control condition when BTX-ion is around. And so this was the killer experiment showing that this BTX-ion can really allow slow inactivation to occur. And so that's how we came up with this first pharmacological master key for slow inactivation. Now we have this compound BTX-ion that can discriminate fast and slow inactivation. And we came up with this model where BTX, just like BTXB and BTX-CHX, can promote activation, so facilitate channel opening and block all the aspects of inactivation. On the other hand, we have BTX-ion that can promote channel opening, block fast inactivation, but lets slow inactivation to occur. And this was very cool because now we have this tool that we can use to study sodium channel gating, and that can also be used as a inspiration and platform for the development of sodium channel modulators. So as Jelena mentioned at the beginning, this is also something that my lab is interested in. I'm showing here the 9 different isoforms of sodium channel with their tissue-specific expression, and these channels are involved in many diseases. They are also the target of many drugs, including local anesthetic, antiarrhythmics, and anticonvulsants. But these drugs are not very good at discriminating different isoforms of the sodium channels. And so we are exploring all the structural and biophysical information we have on these channels and using some new strategies to hopefully develop better sodium channel targeting modulators that can target regions of the channels that are different from different isoforms, and so that we can hopefully get isoform selective sodium channel modulators in the future. For the last part of my talk, the last story I would like to tell you is about the first structural view of ion channel biogenesis. And this is a story on calcium channels, basically. These are voltage-gated calcium channels responsible for many functions, including cardiac depolarization, smooth muscle contraction, skeletal muscle contraction, as well as other endocrine functions such as insulin secretion. And here I'm showing an example of the involvement of these calcium channels in the ventricular action potential, where you can see that the conductance for this calcium channel is responsible for this phase 2 or plateau phase of this action potential. And over 35 years ago, it was shown that these calcium channels actually don't work alone, just like many other channels. They actually work as multi-subunits. And here, what was shown was that this calcium channel require alpha subunit, which is actually the pore-forming subunit, and other subunit, including the beta subunit, alpha to delta, and gamma. And the first structure from Nin Yang's group was actually revealed 9 years ago of the cryo structure of the calcium channel, where this beautifully show that all these subunits are there. This is the alpha subunit, beta subunit on the cytoplasmic domain side of the channel, alpha to delta on the extracellular side of the channel, and gamma. So it was clear from the structure and prior many other studies in the past that these channels need to assemble as multi-subunits in order to be functional. What was not known were the steps that are involved in this channel assembly, and also whether chaperone proteins were involved in this biogenesis and channel assembly. And so most of the work of this for the story I'm about to tell you was carried out by my colleague Zhu Chen, a very talented postdoc. And Zhu expressed and purified at least hoped to express and purify only alpha and beta subunits. And what was interesting is that on the gel, he could see the band corresponding to alpha and beta, but also many other bands that were unexpected. And mass spec analysis later showed that this band correspond to a 9 subunit complex called EMC, which stands for endoplasmic reticulum membrane protein complex. Interestingly, these bands also showed up when you try to express and purify the 3 subunit, alpha, beta, and alpha to delta. So all 3 subunits were there. Alpha is here, alpha to delta, and beta, but all these other bands also appeared. And that was very intriguing, but it was also very exciting because this EMC became very popular in recent years, as it was suggested to help in the membrane insertion of tailored anchor protein. And it was also suggested to play a role as a holdase for proteins that are not fully assembled. And the structure of this EMC was actually solved a few years ago, and it has this characteristic crescent shape on the luminal side of the complex. Now, that was super exciting, and we were excited to see what we could get from this complex, because we're able to see that there was a it was bound to the channel based on the gel, because the channel was purified with the tag on the channel. So if these bands show up, hopefully they are bound to the channel. So here I'm showing some cryo-EM analysis where we're able to see in this sample with only alpha and beta, we're able to see classes with the channel, but also with the crescent shape of the EMC. And this is also true, and this is the nice class also that clearly side view that clearly shows that. This is also the case for the sample with the 3 subunit alpha, beta, and alpha to delta, where we could see classes with the EMC, but we could also see classes with alpha to delta on these 2D classes. Now, here's the nicer view of this EMC-bound calcium channel, where you can appreciate the size of this complex. It's basically a 600 kilodalton complex formed by the channel here in yellow and these 9 proteins forming the EMC bound to the channel. And this is the complex with only alpha and beta co-expressed and purified. Now, in the complex with alpha to delta in there, this EMC bound complex was also present. And there was also the second complex, which is basically the fully assembled channel formed by the alpha subunit, the beta subunit, and alpha to delta here. And if we take a closer look, here's the model of this complex. And when we take a closer look at this, we could see that EMC, or the channel, was binding to the EMC on 2 different sites. And the TM dock, or the transmembrane, at the transmembrane region here between EMC1 and the voltage sensing domain 1 of the channel. And at the cytodock, we have this interaction between EMC2 and MC8 with the beta subunit of the channel. So we call this the cytodock, and this one the TM dock. Now, if we have a even closer look at this interacting sites, we can appreciate that there is a number of interactions involved here. So EMC1, in the transmembrane region of this EMC1, engage in van der Waals interactions with this growth between voltage sensing domain 1, S1, and S2. And these interactions are actually very framed on the top and the bottom by hydrophilic interactions, where we have here a salt bridge between this aspartate from the EMC and this arginine from the VSD of the channel. We also have these cation pi interactions on the bottom, where between this arginine from the EMC and this tryptophan from the channel. What was also interesting is to see that all these residues involved in these TM dock interactions were very conserved among other calcium channel isoforms. If you look at this tryptophan, for instance, engaged in this cation pi interaction, it's all in all the calcium channel isoforms. And this suggests that other calcium channels should be able to engage this TM-dock. Looking at the cytodock, we're able to see that there is also many interactions that were involved between different EMCs and the beta subunit of the calcium channel. And this involved van der Waals interactions between EMC2 and the beta subunit, but also other type of interaction like hydrogen bond and salt bridge network between the EMC8 and many residues of the beta subunit. What was interesting, again, just like in the case of the TM-dock, is that all these residues were also present in many other CAF beta subunit isoforms. And this suggests that other beta subunits can also engage this cytodock. Now, we wonder whether both the alpha to delta and the EMC can coexist on the channel, and the answer is no, because when we superimpose these complexes, we could see like there was a number of clashes in this region here on the luminal here on the luminal or extracellular side, depending where you look at the channel. But if you look at the ER, you can see that in this luminal side, you have these clashes between alpha to delta and the EMC, which means that they cannot be together on the channel at the same time. So their bindings to the channel core are mutually exclusive. And the next thing we did was to basically compare the alpha subunit of both complex, EMC bound and alpha to delta bound, and see whether we have the same thing. And here you can clearly see that there is a number of rearrangements that are involved. When we compare this alpha subunit, we could see that the conformations are completely different. And the most striking ones, there will be where the complete unfolding of the voltage sensing domain number 3 when EMC was bound, and this was folded when the alpha to delta was there. There was also this port domain number 3 in lime here that was completely extracted farther away from the pore. And this actually makes the channel not basically it messes up the pore of the channel. And this is not the case when alpha to delta is there, where the channel is fully folded and the pore is nicely assembled. And so what this means actually is consistent with what I showed you previously, that EMC and alpha to delta do not bind to the channel the same way. So the conformation required for the binding of the EMC is very different from the one required for the binding of alpha to delta. So now, what does this all mean? We try to see whether this binding of EMC actually has any functional relevance. And for that, we wonder whether we could disturb the interactions either at the TM-dock or the cytodock and see if we can have any functional effect on the channel. And so we did some electrophysiology, most of which was carried by a talented electrophysiologist, Sel Zhang. And what Sel measure is here the current density of the alpha and beta. And when Sel measure the current density just with the alpha, we basically kill the channel. And this is not new. It has been shown before that beta subunit is very important for channel expression to the membrane. Now, when we mutated the 3 residues involved in the interactions at the TM-dock, we have a 50% reduction in channel activity, meaning that this interaction is very important for channel functional expression. And mutation of the cytodock, so there are 3 different types of mutations, either a deletion of this loop here, or deletion of the loop plus single amino alanine substitution of these residues, or mutating all 11 residues of the cytodock. And basically, all these manipulations cause a decrease in channel activity. And when we start introducing alanine mutation, we basically kill the channel. This suggests that the cytodoc interactions are very important in channel activity and channel expression. And what happens when we mutate both, when we disrupt both the interactions in the TMDoc and cytodoc, we basically reduce completely channel activity. So what all this means is that all these interactions are very important. And another last experiment was to see whether if we express channel on cell lines that have knockout for EMC, and in this case, we have EMC5 and EMC6, again, we kill channel activity, suggesting that this EMC is playing a big role in channel assembly and channel expression to the membrane, because actually in some cases we could still see some currents even if it was low, but the biophysical properties were the same as the wild type. And so to summarize, this is how we came up with this model, and there are also other data that I didn't show today for the interest of time, but we came up with this model where the calcium channel alpha subunit is made, and when it's on its own, there is this quality control processes that will lead to its degradation, because if it's not fully folded, it will be degraded. And in the ER, the EMC is there to basically hold the alpha subunit while it's being made and it's being fully assembled. So when the channel is not fully assembled, it can be protected by the EMC. And when the beta subunit comes in, it will make this interaction even stronger between the EMC and the channel. And once the channel is fully made and fully folded and ready, then the EMC will hand it over to alpha to delta to form the fully assembled calcium channel. And once the channel is done doing his job, then it will later be degraded. And this is the summary of the story. I want to take 1 minute to kind of highlight also some work in my lab that was inspired by this previous story, where we are interested in sodium channel complexes in health and disease, and has been shown that NaV1.5 can interact with more than 17 proteins. NaV1.7 can actually interact with more than 260 proteins throughout the life cycle of the channels. And we are interested in exploring these complexes and using structural analysis and biophysical studies to understand how these complexes fine-tune channel function. And we're also collaborating with the UCLA Division of Cardiology to assess channel mutations found in patients on how this can modulate sodium channel complexes. And this is the most important slide of my talk, where I would like to acknowledge all the people who are involved in this work. All this work, the work I presented today was done in Dan Minor's lab at UCSF. So I want to thank Dan, who is my mentor, and all the members of the Minor lab who contributed actively on this work. This also involved many amazing collaborations with Justin Dubois at Stanford, Belin Zaro at UCSF, Lauren O'Connell at Stanford, and Jack Dumbaker and the California Academy of Sciences. I'm also grateful to the members of their groups who contributed to this work, and many thanks to the Stanford SLAC Cryo-EM Center and UCSF EM Core, where the cryo-EM data were collected. I'm also grateful to members of my lab who helped me set up the lab and who are really making the lab an amazing place to work at. And a big thanks to funding sources and all these funding organizations that funded me personally. And I'll be happy to take some questions. Thank you for your time.
Thank you very much, Fayal. We have a question, but actually, and Connor as well. But I'll actually first read out the questions from the chat. We don't want to abuse our position. Okay, so the first question is from Jader Cruz. Nice work. Is there any information about the biosynthesis pathway of BTX?
So it is thought, the short answer is no, I don't know, because it's thought that this BTX is made probably in some bacteria, and then beetles will have this bacteria, and these beetles are actually the food that the bird and the frogs eat. So that's how they get the toxin. Information about the biosynthesis itself of the BTX is not, to my knowledge, is not known.
Okay. So the next two questions are from Damien Bell. The first one is, if BTX in P. terribilis is used for predation, why would it evolve to carry sufficient amounts to kill 8 adult humans? Even if evolved as defense to avoid predation, most predators presumably would be small, such as birds and mammals. Small birds and small mammals.
That's a great question. So just to clarify, for on the Phyllobadis terribilis, it's being used to defend themselves against predators. They are not using it for predation. But great question. Why do they need to carry so much of BTX? It's always about it's like an arm race. It's like how much exposure they have to predators, and if they have this actually comes back to the first question in terms of biosynthesis. I don't know how fast they can accumulate this toxin. And so probably my hint was that they just need enough so that they can use it and protect themselves longer, because I'm not sure. Maybe they don't know if they're going to be able to accumulate more of it. But that's a very, very interesting question, unless there are some bigger animals out there that can also eat these tiny frogs.
Okay, and the second question from Damien was, the time course of P. terribilis showed it took tens to hundreds of minutes to recover from paralysis from STX and BTX. So these paralysis recovery time courses are most likely due to saxifilin dissociation concept for the STX and BTX. Is that right? And do the recovery times follow saxifilin STX and BTX KD, assuming these are known and have been defined?
Oh, okay. This is a very, very good question. So the challenge experiment I showed clearly showed that for BTX there is no change with the PBS. For STX and TTX, yes, the frogs wake up much later, and it was mostly true for TTX. And actually, something to mention here is that TTX has isoform selectivity, in the sense that some isoform, like the cardiac sodium channel isoform, is resistant or somewhat resistant to TTX, whereas other isoform are not. So I think the change there is due to the fact that the toxin is targeting some isoform but not the others. So the animal is asleep, but the heart is still beating, and then later on, once you get rid of the toxin, it will wake up. Regarding the dissociation constant and how fast the recovery was in that experiment where I applied saxifilin, was basically the affinity, the KD. We're talking about nanomolar affinity of the channel for TTX, and this is basically the same affinity for of the saxifilin to the toxin. So they have similar affinity, and that would make sense with the fact that with 1-to-1 molar ratio, you would have 50% recovery, and when you increase that by 2, you get 100% recovery of channels. So yes, it does make sense.
Okay. So the next question is from Khalid Udaha. He says, great work, Ali. And the question is, my question is about the voltage-gated calcium channels loss of function mutants. Were all the mutants showing an alteration in the channel expression?
Yes, that's a very good question. So when we look at the biophysical properties like voltage dependence of activation, inactivation, or kinetics, they were the same between the mutant and the wild type. The only change was the current density. And actually, when we assess membrane protein expression, that was the only parameters that was affected, suggesting that the CMC is really playing a big role in channel trafficking, leading the channel to the membrane or helping to fold before it goes to the membrane, and that is not affecting any of the biophysical properties of the channel. Great question.
So the next question is from Jian Hao. Very interesting talk. I'm just wondering if the saxitoxin STX association happens in all the tissues of the poisonous animal. And when other animal get STX from the poison animal, will the saxifilin come together with it? So when other animal gets STX from the poisonous animal, or gets poisoned by it, will the saxifilin come together with the poison, essentially?
That's a great question. So basically, the idea here is that the toxin sponge protein, whether it's saxifilin or batracofilin or any other toxin sponge protein, will be used to first of all, these animals get the toxin from the diet. So the toxin needs to be moved from the stomach to specialized tissues. So these proteins could be used to move the toxin around, but also to protect the animals. Once the toxin is on the right organs, then the toxin is on its own. So on the skin of the poison dart frogs, for instance, you can see only the BTX is there. So when we eat, or when an animal eats or touch it, he gets only the toxin, but not the protector, not the toxin binding, the toxin sponge protein. And to the first part of the question, whether the interaction, the association happens in all the tissue of the poison animals, I think it happens only in the blood and during the transport mechanisms with the toxin. Once it's done, the animals doesn't need the protector on all the tissues, unless there are some sodium channels in there where they would need the protector.
Okay, so the next question is from Sam Brown. Great work. Did you perform any current clamp with BTX? I'm curious if you saw increased firing with increased slow-in activation and subsequently increased persistent current, or if it would reduce due to the decreased transient/fast inactivated current.
That's a very interesting question. So I did not do any current clamp with BTX, but the way this toxin works is it binds inside the inner pore of the channel, and it basically prevents the channel from inactivating. And so my guess is that once the toxin is there, you basically have an open channel. You cannot fire several times. So yes, the expectations that you would see, instead of seeing actually an increased firing, you will just kill the firing at all, because there is no new firing, and you wouldn't see any new action potential happening in this. But I did not do current clamp experiment on this. Good question.
So one last question in the chat is from Guy Salama. Is B-catenin acting as a chaperone, or does more in terms of modifying the channel function?
So I did not talk about B-catenin. I'm not sure what the question is referencing to, but if this is talking about the EMC that I mentioned, then the idea here is the EMC is playing a role of a chaperone or a hold days to basically to hold the channel and wait. And actually what's interesting in this is that the binding involved the voltage sensing number 1 and the segment number 1. So that's the very first domain that's made of the channel. So the idea is that as soon as the channel is starting being made, the EMC is there to catch the very first segment and wait for all the other segment to be made and fully folded before you can hand it over to the alpha to delta.
Okay, and one last question from Connor.
Thanks, Yelena. Thanks, Fayal. That was a fascinating talk in many different directions. I know you're working overtime now. I'll try and be quick. So I was wondering, in addition to your demonstrated effects on reducing plasma membrane expression when you knock out the EMC complex, I wonder if the EMC complex is actually preventing any activity of the calcium channel when it's in the ER, right, which could be pathological. So if you knock out the EMC, do you see changes in ER calcium levels or cytosolic calcium levels when you overexpress the CAV channel?
So we did not look at that. That's a very interesting point. We did not look at the ER calcium level, but something important to mention here is that when EMC is bound to the channel, the pore is completely messed up. You have the pore domain number 3, which is extracted. You have the voltage sensing domain number 3, which is completely unfolded. So basically, the channel is not functional. And we think that this is also a mechanism to protect the ER, because if you have a calcium channel that's functional there, you're going to have a a change in calcium homeostasis, and that would be problematic for the ER. And so we did not measure that, but we think that based on the conformation of the channel, when it's bound to EMC, it cannot coordinate any calcium ions through, and it cannot cause all those problems.
Yeah, that's really cool. It's something I'd always wondered about. How do we regulate channel activity during the trafficking process there? That's great.
Yes.
Thank you.
Yes. Yeah, pleasure.
Yeah. That's great. Thank you so much, Fayal. Thank you, everybody, for coming. We're just 5 minutes over, but we started a little bit late. Okay, and the next seminar for May is Claudia Moreno talking about the heart rate adaptability. Okay, thank you.
Exciting.
Thanks, everybody.
Thank you, guys.